Hello, my dear students. I'm Dr. Vaishaly Bharambe. I've been teaching anatomy for more than 25 years and I love it. So, today we are beginning the cell series, a lecture on the cell. So, what is cell series all about? We're dividing this into cell part 1, part 2, and part 3. What are each of these parts of the cell all about? Let's understand. Cell part 1. In cell part 1, I'll be introducing the cell to you and I will be discussing about the nucleus and the plasma membrane. Okay, so what will we discuss? I may ask you to draw and label the cell beforehand. Okay, see, you have been learning about the cell right from your school years. So I will ask you to maybe draw the cell, okay, out of your memory. Then understand that the cell has a cytoplasm and the nucleus. Then we will discuss the nucleus in detail. What is the nuclear envelope, nucleoplasm, nucleolus, as seen here. And then we may discuss how the chromosome slowly unravels to form euchromatin or heterochromatin. It keeps on uncoiling the chromatin fiber till you have a DNA. We will discuss that in brief. Then I will discuss with you how a cell is divided into cell organelles are divided into membranous and non-membranous, which help to divide the cell into microcompartments. We will go on to talk about the plasma membrane, which is made up of phospholipids, carbohydrates, and proteins, okay, and how they control the entry of material into the cell, as in endocytosis, or outside the cell, as in exocytosis. Throughout all this, I will be using a lot of images to help you understand. At the end of cell part 1, there will be a short quiz, which is cell part 1 quiz. I will expect you to solve this quiz before you come to cell part 2. Throughout this, I ask you a lot of questions in my lectures. The answers to all those questions are given in the form of notes. So if you do not get your answer in the lecture, please go to the notes. Sometimes some part which is not discussed in the lecture, okay, may be elaborated further upon in the notes so that, if in case you are interested, you can read that and further enhance your knowledge. Cell part 2 is all about cell organelles, okay, such as now the mitochondria and its various parts and functions. Mitochondrial inheritance, a very peculiar form of inheritance where the mother can pass the disease to her children, son and daughter, but the son cannot pass the disease to his daughter and son. What is this? How is it? Why is it? That's what is mitochondrial inheritance. Then we'll talk about the protein producers of a cell, the ribosomes, endoplasmic reticulum, which is tubules, vesicles, or sacs of two types, rough and smooth. How the rough endoplasmic reticulum is found in high quantities in protein-producing cells such as pancreatic acinar cells, or smooth endoplasmic reticulum found in cells that produce lipids and steroids. Then we'll discuss Golgi complex and parts of Golgi complex, cis, medial, and trans Golgi. Then we will discuss also how a rough endoplasmic reticulum releases protein through transport vesicles, picked up by cis Golgi, passed through to trans Golgi, released as secretory vesicles, which fuses with plasma membrane to release the material outside by exocytosis. How does this happen in a cell? Then we will talk about peroxisomes as well as lysosomes. Okay? And what is lysosomal storage disease? Remember, deficiency or malfunction of specific lysosomal enzymes result in accumulation of substrates in lysosomes, resulting in lysosomal storage diseases. Then again, test yourself with the help of a quiz at the end of cell part 2 and a few notes for additional knowledge. Cell part 3 is about cytoskeleton and cellular projections. So, cytoskeleton consists of microtubules, microfilaments, intermediate filaments, which should be discussed. The structure of microtubules, 13 protofilaments joining to form a microtubule, microfilaments forming the core of microvilli, centrioles, which are surrounded by MTOC, What is MTOC? Find out in the lecture. And how this MTOC releases microtubules, which are utilized by the cell for various purposes. A brief diagram development will be done by drawing the section of a centriole. I may also ask you to draw a cell at some point along with me. I have a method in which I get a lot of drawings done in my class. So I will be putting up video of how to draw a cell in case you're not able to do it on your own. I will release a video on how to draw a centriole to help you draw the centriole. Then how the cytoskeleton helps to support cellular projections such as cilia and microvilli, structure of a cilium, how within the cilium microtubules are held together by proteins such as radial, spoke, nexins and dynein arms, and how an abnormality in this results in various clinical conditions. Differences between flagellum and cilium, differences between microvilli and cilia. This will be the end of our cell series, okay, which will again end with the help of a practice quiz, cell quiz part 3. Again, there will be some notes given there so as to help you to add on to your knowledge, and maybe a video to help you to understand how to draw. So I'm really looking forward to this journey with you when you travel through the cell with me. Okay, begin the cell series now with your first assignment. Draw and label a cell from your memory. You will draw it from your memory. You will not look at any internet image or any school book image to draw. Just draw whatever you remember of the cell as it was taught to you in your school years. Okay, and armed with that visual image, let's begin our journey through the cell by attending cell part 1. So I'll see you in the first lecture in the cell series, cell part 1. Bye.